Hello, everyone, and welcome to the Anti-Doping Podcast, brought to you by the Partnership for Clean Competition. I'm your host, Dr. Marie McNeely, and today we are joined by our guest, Dr. Olivier Salamin. Listeners, Olivier is the operational manager of the Lausanne Athlete Passport Management Unit, or APMU, within the Swiss Laboratory for Doping Analyses at the Lausanne University Hospital. And the hospital is known in French as the Centre Hospitalier Universitaire vaudois, CHUV, CHUV. So listeners, you may hear us refer to it that way throughout our conversation today. And if you need a refresher, listeners, on the Athlete Biological Passport, or ABP, I recommend you take a moment to check out episode 131 with our guests, Dr. Reed Aiken and Norbert Boom, where we did an ABP deep dive, because today we are looking forward to discussing some of Olivier's research on the discovery and validation of biomarkers to detect prohibited substances and methods, particularly related to the Athlete Biological Passport. So, Olivier, welcome to our show today. How are you?
Hello, Marie. Thanks. I'm very good. Thanks for having me. It's a great pleasure for me to be part of this PCC podcast.
Well, we are so excited to welcome you to the show today and to learn more about you and the work that you do. And perhaps to get us started, Olivier, can you tell us, how did you initially get interested in anti-doping science?
I've been always fascinated by doping stories and scandals. Since I was a child, though, I've never been quite sure why. It's a bit funny, but I still remember the Festina affair quite clearly, even though I was only 6 at the time. Especially how a French TV show mocked the whole situation of this Festina scandal. Later, a course on doping and anti-doping in high school deepened this interest that I already had in anti-doping and doping in general. And then when I discovered that the only Swiss anti-doping laboratory was based in Lausanne, actually not that far away from where I am from, I saw it as a unique opportunity to get involved in this field once I started also the university.
Oh, very cool. And I'd like to talk a little bit about the research that you did in your early years, particularly your PhD research at the University of Lausanne that was in collaboration with the Swiss Laboratory for Doping Analyses that you mentioned, and also the Center of Research and Expertise for Anti-Doping Sciences, also known as RED. So can you tell us, Olivier, how did this unique PhD opportunity come about?
So this opportunity arose just as I was completing my master's degree, which I already was conducted at the Swiss Laboratory for Doping Analyses. And that same year, the Center of Research and Expertise in Anti-Doping Sciences, as you call REDS, was actually established at the University of Lausanne by my former director, Martial Soji. And this new center created an opening for a PhD position with the agreement that all the analytical work would continue to be carried out at the Swiss Laboratory for Doping Analyses. So, of course, when I found out about this opportunity, I showed my deep interest to Martial Soji, first by staying at the lab as a trainee after my master until the PhD position was open. And then I was chosen and I could start. So the path was quite open for me, and that was a real nice opportunity.
I love it. And it sounds like you took some steps sort of proactively to make sure that opportunity was open and that you could take advantage of it, which I appreciate. Now, you mentioned that you've always kind of had this passion for anti-doping. Were you an athlete yourself in your early years?
I don't know if I can call myself an athlete, but I'm very passionate about sports. So when I was younger, I was doing karate, football, and now I'm also into cyclism and a lot of different sports as workouts, paddle tennis, etc. So I've been interested in sport my whole life, and combining this interest in sports with biology, which is also a real big interest for me, was like the perfect opportunity. And I think that's probably what most of people that are into anti-doping that would say this.
Definitely. And now let's go into a little bit more detail about this cool PhD research. Can you tell us, what did you focus on?
So given the Lausanne pioneering role in the development of the Athlete Biological Passport, which I will name ABP now, so my PhD research focused primarily advancing the ABP by exploring the potential of various biomarkers, primarily measured in blood. So blood was my big focus as a matrix, using various omics approaches, as we call them.
Very cool. And Olivier, I know you did pioneering work on transcriptomic biomarkers among the omics for blood doping. So can you maybe briefly explain what transcriptomic biomarkers are for our listeners who might not be familiar and how they can be used to identify blood doping?
Transcriptomic biomarkers are mainly indicators that are based on gene expression levels. So essentially, they reflect which genes are turned on or off, so stimulated or suppressed in response to a specific condition or stimulus. So in other terms, they represent the number of transcripts, so the messenger RNA expressed by a specific gene. And in the context of anti-doping, they can provide a snapshot of how the body would respond at the molecular level to blood doping practices such as erythropoietin EPO use or blood transfusions, for example. And interestingly, the reticulocytes, so the most immature red blood cells in circulation, still contain some residual RNA, which is still functional for the maturation into red blood cell. And this RNA contained in the reticulocyte can provide quite an accurate reflection of a possible altered erythropoiesis, so the metabolism that is linked to the production of red blood cell by the bone marrow.
That makes sense. And can you maybe give us a little bit more detail about some of the kinds of transcriptomic biomarkers that you looked at in your PhD work?
So one of the most studied transcriptomic biomarkers in anti-doping is what we call ALAS2. So this gene encodes for ALAS synthase, an enzyme which is involved in heme metabolism. And heme is a key component in hemoglobin production by red blood cells. It means that changes in ALAS2 expression can reflect alteration in erythropoiesis, a common target in blood doping strategies. And we also have other examples of transcriptomic biomarkers, which include CA1 RNA transcript. So CA1 encodes for carbonic anhydrase 1, an enzyme which is involved in gas exchange within the red blood cells as well. It has been shown that this gene can be modulated as well by EPO administration or blood transfusion, making it a potential marker also of blood doping. And finally, our lab has also investigated another gene, which is called SLC4A1 as a transcriptomic biomarker. And this gene encodes the anion exchanger, which is a membrane protein that plays also a key role in maintaining the acid-base balance in red blood cells. So all those genes were always related to red blood cell metabolism, meaning that blood doping would also alter the expression of those genes.
Very interesting. And I know your PhD research, you also performed a fascinating administration study with testosterone gel, working with healthy women subjects. So can we talk a little bit more about this study and perhaps some of the challenges that you encountered in just organizing and getting the study started?
Yeah, I think that this study is probably the one that I'm the most proud of. I think that the first major challenge that we faced with this administration study was first obtaining the ethical approval. Because you can think that administering testosterone to women required quite a thorough justification, and we had to ensure also that the potential risk of side effects were minimized through careful consideration of the dose first, but also the treatment regimen. So this approval process took nearly a year, so quite a lot. And also the second challenge related to this study was recruiting the suitable volunteers, specifically because women who were not only willing to take testosterone, but also not using hormonal contraception and who had regular menstrual cycles. So that was also a big challenge. And finally, maintaining the participant engagement throughout the study was also an additional difficulty because the volunteers were required to visit the clinical trial unit nearly every other day for a period of almost 3 months. So quite a heavy protocol. I had to make sure that they would still comply with the study design.
Definitely. And how many participants were you aiming to recruit for this study?
So at the end, I could recruit 14 female volunteers. And what we did, let's say, to ease the whole organization of the thing, that we divided the engagement between 2 periods of time. So we started first with 7 volunteers for 3 months, and then we started again with 7 other volunteers for the 3 remaining months.
That makes sense. Yeah. I think having 14 people come in every other day would get kind of chaotic. So
exactly. For the nurses, it was already quite a hard job. So we didn't want to make the job harder than it was already.
Certainly. So then what were the findings of this study?
This study could demonstrate that the sensitivity of the urinary steroid profiles was quite limited in women, and it was primarily due to menstrual cycle related fluctuation, especially in epitestosterone levels, which is used in the testosterone on epitestosterone ratio and on the 5-alpha-diol on epitestosterone ratio, which are used as marker in the urinary steroid profile, and also the generally quite low concentration of steroid excreted in urine of female subjects. And in contrast, we could demonstrate that steroid profiling in blood, so in serum and particularly for testosterone, showed quite a markedly improved sensitivity. And in addition, we also introduced the testosterone to androstenedione ratio as a novel biomarker for longitudinal monitoring in women. We could show that tracking this ratio significantly enhanced the ability to detect testosterone gel administration in women. And finally, using a subset of samples, of urine samples that we collected throughout the study, we also observed that isotope ratio mass spectrometry, RMS, did not consistently as well provide sufficient sensitivity across all subjects. So taking together all those findings could really help to understand how sensitive we were related to the detection of testosterone administration in women.
That is remarkable. And I think the study in particular really generated valuable data on steroid profiles in women, and the development of this testosterone to androstenedione ratio, the T to A4 ratio, as some listeners may know it, was a big step in the field. So Olivier, can you talk about how this study helped you for your career?
So yeah, of course, I mean, you mentioned first the implementation, or let's say the introduction of this ratio, but I will start first by saying that for this study, as I was involved in every stage from grant writing and obtaining ethical approval to subject recruitment, sample collection, and reporting and analysis. So this comprehensive involvement allowed me to gain quite valuable insight into every aspect of conducting an administration study, so particularly in terms of project management. So I have to say that first, this study really helped me in growing into this aspect. But as you said, I also developed quite significant expertise in interpreting steroid profiles, especially blood steroid profiles in female athletes. And it was especially rewarding to see that a few years later, so in 2023, the blood steroid profile was officially implemented in the athlete biological passport, including this specific ratio.
I think that is remarkable. So if you had to summarize, Olivier, what were just some of the key findings from your full portfolio of PhD research?
As mentioned earlier, one of the key findings from this PhD research was the demonstration of the advantages of blood steroid profiling over urinary profiling in female athletes. So that was the first thing. But I have to say that I'm also proud to have contributed to the early investigation into the transcriptomic biomarkers for the detection of blood doping, mainly related to these specific genes, ALAS2.
I think that is wonderful. And then you finished your PhD, of course, and went on to do a postdoctoral fellowship focused on metabolomics. So for listeners who might not be familiar, can you explain what metabolomics is and how it's relevant for anti-doping, Olivier?
So metabolomics is part of all those various omics approaches. And we can define metabolomics as the comprehensive study of small molecules known as metabolites within biological samples such as blood or urine. And these metabolites kind of reflects the physiological state of the organism and can provide as well valuable insight into metabolic pathways and changes in response to various stimuli and in anti-doping, including drug use or physical stress. So in the context of anti-doping, metabolomics offer quite a powerful tool to detect subtle physiological changes that could be induced by doping substances or even methods such as blood transfusion, even when the substances themselves may no longer be detectable. So that's one of the main advantages. So, for example, certain metabolic signature of pathways may be disrupted or upregulated following the administration of a performance-enhancing drug, and metabolomics can help to capture those changes. So it holds quite a great potential as a complementary approach to traditional analytical methods, and especially uncovering new biomarkers of doping.
Definitely. And I think every approach that you use has pros and cons. So, Olivier, if you had to kind of map out what are some of the potential benefits and maybe the limitations as well when it comes to using metabolic biomarkers or these signatures that you see in anti-doping?
Metabolic biomarkers, as I said, they offer several promising benefits. And one key advantage is their ability to capture the physiological effect of doping substances or methods, because they represent the very end of the chain of those omics approaches. So, you have genomics, transcriptomics, proteomics, and finally metabolomics. So the metabolome, which represent the very end of this chain. So they have this ability to capture the effect even when the substances are no longer directly detectable. And this is particularly useful for identifying indirect markers or patterns associated with drug use, such as altered metabolic pathways or stress response. So metabolomics can also support the development of longitudinal monitoring strategies. So the goal is to find new biomarkers that could be integrated into the athlete biological passport. So by tracking subtle but consistent deviation in an athlete's metabolic profile over time. So however, we still have some limitation to consider using metabolomics. And many past studies have employed what we call untargeted metabolomic approaches, which can identify the wide range of candidate biomarkers. While this is useful for discovery, a major issue using this approach has been the lack of validation of those candidates across those studies. And additionally, metabolomic profiles can be influenced by a variety of confounding factors such as diet, exercise, circadian rhythm, or even individual variability, which can then also complicate the interpretation of longitudinal monitoring. So when using a metabolomic approach to investigate potential new biomarkers, these confounders need to be taken into consideration as well.
I see. That's really helpful for you to kind of lay it out for us. And I know you bring these tools of transcriptomics that we mentioned from your PhD work and these now metabolomics tools from your postdoc work to your current position. So let's talk a little bit more about what you've been up to for the past year or so, Olivier. Perhaps first we'll take a step back. How did you find out about and decide to pursue this position at the Lausanne Athlete Passport Management Unit?
Sure. So during my postdoctoral period, I remained quite in close contact with the Lausanne laboratory, especially with Tia Kuran. And when the blood steric profile and the endocrine modules were being implemented, the need arose for someone to oversee the full scope of the APMU activities and operation in Lausanne. So basically, they reached out to me for this role. And given that the athlete biological passport had been the central focus of my PhD research, this represented a unique and exciting opportunity to apply my expertise in a practical and impactful way as well.
I think that's wonderful. It sounds like the position was almost made for you.
Maybe.
Now, can you tell us, Olivier, a little bit more about the Lausanne APMU or Athlete Passport Management Unit?
So the Lausanne APMU has been at the forefront of the athlete biological passport since its implementation, basically since its introduction. And Pierre-Edouard Sotas, who was working at the Lausanne laboratory at the time, basically developed the adaptive model that is used within the ABP together also with Marcel Saugy. And as a result, the Lausanne APMU has been active for many years with its operational activities beginning as early as 2008, if I'm not mistaken. And currently we are 11 people that are actively involved in the routine operation of the Lausanne APMU. So we have also lab technicians that are also involved into the daily interpretation of the profiles. And the overall purpose of an APMU is to evaluate an athlete's passport or profile when a new sample has been added to this profile. And the APMU is responsible to assess the validity of the sample first, to do an interpretation of the new values in comparison with athletes' history, and to provide also recommendation for further testing or additional analysis to the passport custodian, which is the owner of the passport. In Lausanne APMU, we basically assess all three modules, so hematological, steroidal, and endocrine module from various national anti-doping organizations, as well as major international federations. So the variety of sports and discipline is quite broad, and it's very interesting to see some patterns that are also specific to different sports, which also altogether increase the expertise that is required for the interpretation. So this is basically what we are doing at the Lausanne APMU.
Excellent. And then to maybe give us some insight into a day in the life of Olivier managing this APMU, what are your roles and responsibilities there as operational manager?
So as the operational manager of the APMU, my role is first to oversee the activities across the different modules of the athlete biological passport. So namely the hematological module, the steroidal module, and endocrine module. So a key part of my responsibility is to ensure the consistency and also the harmonization in how we can provide recommendation to the passport custodian, regardless of the module. And I also work to promote a more integrated and holistic approach to interpretation, so encouraging the crosstalk between the modules to improve the overall assessment of an athlete biological profile. And in addition, I provide scientific guidance and support to the team of people that are involved in the daily evaluation of individual passports, which ensure the robustness and the scientific integrity of our evaluations. So overall, it is both challenging and rewarding to be involved in all three modules, as it requires quite a solid understanding of the diverse physiological processes, including erythropoiesis, steroid metabolism, and growth hormone regulation.
That makes sense. And I know not all WADA accredited anti-doping labs have these athlete passport management units or APMUs. So can you talk a little bit more about how the work done at the APMU complements or maybe adds to the routine work that's being done at the WADA accredited Swiss Laboratory for Doping Analysis?
You are very correct. So first, it's important to note that only anti-doping laboratories are authorized to host APMUs, but not all of them have APMUs. So in Lausanne, we try to make a clear distinction between the activities related to the laboratory and those related to the APMU. So this is why we have separated operational managers for each of those activities. And the work done at the laboratory plays a crucial role first in supporting the routine operation of the APMU. So for instance, biomarkers values can sometimes be influenced by analytical factors. So it's essential for people evaluating the profile to have a deep understanding of the measurement methods to ensure also a proper interpretation. And the APMU's primary goal is to detect doping indirectly. So it kind of complements the laboratory focus on directly detecting the prohibited substances. So together, both approaches create an additional layer of monitoring. And this synergy not only complements, but also enhances the laboratory works, leading to more accurate detection and also more effective prevention of doping in sports.
Well, that makes sense. And then thinking about, I guess, the research side, maybe separate from the routine work side, why is it important to continue to be working on and developing new methods and biomarkers to detect doping?
I believe that the athletes, and probably especially their entourages, are quite well informed about how anti-doping methods work, and they are probably aware of the substances that remain challenging to detect. And I think this is particularly true for endogenous substances that can be taken as doping substances like testosterone, for example. Since direct detection of such compounds is not feasible due to the need to differentiate between endogenous production and exogenous exposure, it's crucial to continue developing new approaches and exploring additional biomarkers for those substances. And these complementary biomarkers could not only improve the overall detection, but also aid in a way in the interpretation of profiles when used in conjunction with the existing biomarkers already implemented in the ADP.
I see. And for you managing the APMU, can you tell us a little bit more about how you balance the routine work and your responsibilities in evaluating all of these passports and also being able to do this work on research and development as well?
Most of my job is mainly related to the APMU activity, but as you said, I still have a percentage of my time dedicated to research. But I'm also, when I'm evaluating profiles daily, I'm always thinking about the possible opportunity that can arise when you see something specific in a profile. So for example, sometimes you may think that there are some confounding factors that it seems that you see an impact on the markers, but when you check on the literature, there are not so many things related to the possible impact of those confounders. So it kind of also generates some ideas.
Oh, that's nice. It sounds like the routine work informs the research, and then the research also informs the routine work.
Yeah, exactly.
And we know that the 3 ABP modules focus on detecting blood doping, administration of steroids, and also administration of growth hormone. So what are some of the promising new biomarkers of doping, Olivier, that you are particularly excited about right now for any of these 3 modules?
First, I would say that our lab has shown that serum dihydrotestosterone, DHT, could serve as an additional marker, particularly for detecting transdermal testosterone administration. However, this approach is currently limited by the low concentration of DHT and its relatively low analytical sensitivity. But at the moment, I'm also involved in a project exploring the potential of stearic sulfates, so phase 2 metabolites, as supplementary markers, both in urine and serum for the detection of testosterone.
Excellent. And I know you have a lot of different things going on right now. Is there a particular project that you're working on that you would like to tell us more about that you're excited about today?
Yeah, as I said, in addition to the project exploring the steroid sulfate markers, I'm also working on a project investigating candidate biomarkers for EPO administration, but this time using targeted metabolomics. So through this approach, I analyzed samples previously collected during an administration study and could identify 2 candidate biomarkers that were quite responsive to stimulated erythropoiesis. And these metabolites were part of the purine metabolism pathway. And this approach was particularly valuable as it provided a high level of confidence in the identification of metabolites, in contrary to untargeted metabolomic approach. So I can now move forward with confirming these preliminary findings by developing quantitative methods for these 2 compounds.
Very interesting. And you mentioned this sort of dichotomy. This is a targeted metabolomics approach versus an untargeted approach. How do you know what to target?
So basically, when you say targeted, you try to select some, let's say, cellular processes or metabolic pathways that are of interest and quite well known and quite well characterized as well. So once you have selected those different pathways and cellular processes, what you need to do is that you need to acquire the standards of all the different metabolites involved in those pathways and then set up an analytical method injecting those standards into the mass spectrometer, acquiring the identification criteria and the retention time for those metabolites. And then you can start analyzing your real sample. And since you had the standards for those metabolites, you're pretty sure that what you are measuring is the metabolite itself. And you don't need any bioinformatical software or workflows to try to identify the metabolites that you're seeing in your sample.
That makes sense. And thinking about the future, if these 2 candidate biomarkers for EPO administration pan out, what are the potential impacts of this work?
If I can make it through the whole process, I hope that if at some point it's included into the ABP, it could help for the detection of EPO administration and also helping to do interpretation of hematological profiles in relation to the ones that are already existing markers, such as reticulocyte percentage, for example, which is quite sensitive to EPO administration.
Makes sense. And I know it's not easy getting to this point where you're ready to translate your research into actual practice in anti-doping. So, Olivier, can you give our listeners some insight into what are some of the barriers that you face in this translational process?
Yeah, there are many, I would say. And one of the key challenges in translating new biomarkers into practice is the need for thorough validation. So the process can be very long. So when you start identifying potential biomarkers, it's essential to confirm this finding through additional administration studies first, involving also different doses, but also regimen. Then you need to also evaluate the intra-individual variability of those markers because they will be evaluated longitudinally. Because if this intra-individual variability is too important, it will be complicated to discriminate between the natural variability, but also possible doping. However, I would say that the major limitation lies also in evaluating the impact of potential confounding factors, so external elements that can influence the values of a given biomarkers. So that said, many of the biomarkers already implemented in the ABP are also influenced by confounding factors. So while it's crucial to consider these influences, they should neither be deterring the investigation of promising candidate biomarkers.
Definitely. It sounds like they sort of just inform the interpretation later down the pipeline.
Exactly. So if we know their impact and we know how much they can impact the possible biomarkers, it's already a good start. And taking into account this information when we do the evaluation, it's easier.
That makes sense. So I guess if you're thinking optimistically, about how long does it take to get this sort of discovery of a new biomarker to the point of practice, actually that implementation of it into practice?
Oh, it can take years because all those steps requires quite a lot of samples to analyze. So it's quite hard to give a specific time window to go through this thorough validation. But yeah, as I said, it will take years. But once we have already a robust method to quantify those markers, it's already a good start. And then once we have assessed the variability intra-individually, probably the influence of the confounding factors can also be learned while they are already implemented. And I think that's also the case for the blood steric profile at the time. So they were implemented in 2023. So it's been 1.5 years of implementation, and we are still discovering potential confounders that can influence their concentration. So this should not slow down the implementation of a candidate biomarker.
Definitely. And I think another challenge when you think about getting it into practice in labs around the world who are all WADA accredited laboratories is just this idea of making sure everybody's doing everything the same, getting the same results, and getting people to implement the measures to actually use them in their daily practice in the laboratory. So can you maybe give some insight into how that process works?
Yeah, of course. I think this is a challenge because, of course, all labs in the world can receive samples of one athletes, and the value that will be generated from this sample will be integrated into the athlete's profile. So we need to make sure that there is no bias between the laboratories. So we need to have harmonized methods. And in order to facilitate the life of the laboratories, so if we want to implement new biomarkers, we need to also have quite easy new methods to implement. And we need to make sure that those methods are not a challenge for the laboratory to develop. So it's also one aspect that needs to be taken into account is trying to develop easy methods to implement by the lab, because the lab, they already have quite a lot of different methods. So adding always new methods is also a challenge for the anti-doping laboratories.
Definitely. And I think collaborating and working together is key for translating some of these new discoveries in anti-doping into actual anti-doping practice. But it's also just key for everyday life, I think, in the field of anti-doping. So, Olivier, can you talk about how you collaborate with other stakeholders, whether it's funding organizations, the WADA accredited laboratory that we mentioned, and then just others in the anti-doping field in the work that you do every day?
Yeah, so in my role within the APMU, I try to maintain quite close contact with various stakeholders, but particularly the passport custodian, because they are the owners of the athletes' passports. So this often involves responding to urgent requests or clarifying recommendation we provide, or even providing specific training on different aspects of the biology of steroids, of erythropoiesis, something like this. So when it comes to funding organization, there are limited options for anti-doping research. PCC, however, provides an excellent opportunity to support research initiatives. So I'm quite grateful for the chance to collaborate on projects with their backing, which has been quite instrumental in advancing our work, but also related to the collaboration with other laboratories. So in my role, I mainly collaborate with other APMUs. So it's always quite interesting to see other APMU's points of view, to know how they deal with the routine operation, their way of interpreting specific fluctuation that we can see and that they can see as well. And some passport custodians are our clients, but they can be also clients of other APMUs for different modules. So sometimes we also organize specific meetings to share steroid profile with hematological profile of the same athletes with another APMU, just to see if we can have a synergy between the different modules. So as you can see, it's quite complete. We have quite many contacts with the different stakeholders. And I think this is key to make sure that everybody is on the same line and understand also our recommendation and the request that we also receive in return.
Certainly. And I know we were able to recently convene quite a few of these anti-doping stakeholders in one place at the PCC's conference. So you recently attended the conference and you were actually one of the speakers. So Olivier, can you comment on what your experience was like at the event?
So yeah, it's related to this previous question. And I have to say that my experience at the recent PCC conference was excellent. And I was quite thrilled to have the opportunity to be part of the event with this presentation and to share my expertise on blood steroid profiling, especially in New York City, the headquarters of the baseball league. And I have to admit that I was very impressed by the quality, but also the diversity of the presentation with different stakeholders, and was incredibly insightful to exchange ideas, not only with scientists from anti-doping laboratories, but also with individuals from federation and anti-doping organizations. So I think this added quite a layer of multidisciplinary perspectives, which I found to be both valuable and very enriching.
Definitely. And what were your thoughts on the inclusion of the athlete perspective in some of the presentations from former Olympic athletes?
I think this is really interesting because when we work at the lab, we never consider those perspectives. So we receive samples, we analyze the samples, we have the data in the ADP, we interpret the data, but we do not necessarily think about how stressful all this environment can be for athletes. So I think that was really insightful as a laboratory scientist to get those different, let's say, feelings from athletes.
Well, I'm glad you found the conference valuable. Again, we appreciate you presenting your exceptional research at the conference. And you've, of course, been able to do some remarkable research over the years, but I know there are limitations, unfortunately. So if we took away the things that normally hold you back, Olivier, whether it's funding, time, feasibility, whatever the case may be, is there a particular problem in anti-doping that you would most want to solve?
Probably, in my view, one of the major challenges in anti-doping is the limitation on testing frequency, often due to the high costs associated with sample collection, but mostly shipment for blood samples. And additionally, there can be significant delays between sample collection, analysis, and the entry of the results into the ABP profile, sometimes taking days or even weeks. And this time gap, can prevent the detection of emerging patterns or suspicious behavior in a timely manner. So if I had unlimited resources, one of the problems that I would focus on is finding a way to enable more frequent and cost-effective testing with faster results. So ideally, this would involve a system that could provide near instantaneous data, such as probably a sensor, which could allow for more regular testing and real-time interpretation by the APMU, which could help to detect suspicious patterns probably more promptly.
Well, I think this is definitely a big problem in anti-doping, and I appreciate you taking the time to think through what the solution may look like and giving our listeners something to think about. And if our listeners are intrigued by the work that you described today and they would like to learn more about you and what you do, Olivier, what is the best way for them to do so?
People could find me either on LinkedIn, ResearchGate, but if they are more interested in my research than myself, probably they can find my work easily on PubMed or Google Scholar.
Excellent. Well, listeners, definitely get connected with Olivier. Take some time to learn more about his recent research papers and the great work that they're doing there at the APMU. And Olivier, it's been wonderful to chat with you today. Thank you so much for joining us on the show.
Yeah, thanks for having me and thanks for all those interesting questions. And I hope that I could provide some insights of what I'm doing as APMU manager, but also as an anti-doping researcher.
We appreciate it. Thank you again so much for sharing your expertise with us today. Listeners, it's been wonderful to have you here with us as well. We hope you can take a moment to subscribe and rate and review our podcast on your favorite podcast platform. And listeners, please note that the Anti-Doping Podcast features a wide variety of clean sport experts, and episodes may include statements and or viewpoints that do not necessarily reflect the views or values of the PCC. Thank you, and we look forward to seeing you on the next episode of the Anti-Doping Podcast.